author of more than 400 scientific publications and 6 patents. Is this right? Okay. His presentation today titled Decades of Innovation in Nanotechnology in Agroecosystem.
Thank you very much. I have to tell you, I have some patent really, but I have 2 grandchildren as well. And if I have to tell a story to the grandchildren, generally I start like that. Once upon a time, there was something. And I'd like to do the same with you. You look a little bit older than my grandchildren, at least in the first row. But I'd like to talk about 2 fairy tales, but real stories in the nanotechnology. I don't want to talk about general nanotechnology because all of you know the interest and importance in that. But I tell you real stories. And the first story started 15 years ago, or a little bit more probably, when a researcher from Egypt visited my lab. His name is Mohsen Zomara. We will meet the day after tomorrow. My son made a sketch on him on the left side. I was younger at that time. And his topic was food chemistry and food science. He used to deal with producing yogurt and yogurt type of food. And my interest in that time was the selenium and the speciation of selenium. It was a serious problem in my country anyway, especially next to my town. You know, we have a very alkaline soil, and we have a very high shortage of selenium there. So if we have grazing sheep, the young sheep, without treatment, they die within some weeks. And they got an illness which is caused by the shortage of selenium. It is called white muscle disease. They have a pain in the back, they move a special way, and after 2 weeks they die. So the only cure for this is the selenium injection. But of course it would be much better to find the selenium supplementation for them. Of course we can use inorganic selenium, what is mined, to add it simple, but the situation is not so clear and simple. Because this type of selenium is easy to overdose. So if we go to Canada, in Canada they have very high selenium content in the soil. And it can make poisoning for the cows. So you have to find such a selenium form which is not toxic, but it has good bioavailability and could solve this problem. So we tried the yogurt and we wonder what's happened if we have a microorganism like a lactobacillus, how it can convert the inorganic selenium. We supposed we will have organic selenium compounds, but it was not the case. My friend, the professor, brought me a tube like that, and he showed me it is red. It became red. I said, okay, I know it is red elemental selenium. But I started to thinking, if the bacteria produce it, clearly, it should have a special structure. So I decided to make some electron microscopy of that. So that on the left side, you see the bacteria, and we digested it, and finally we got very small and fine balls. Actually, it is nano size, 200 nanometers. Every bacteria, what we tested, produced a different size, but the same size was produced by every bacteria. Why it was interesting? You see the bacteria inside the transmission electron microscope. We had more balls in one bacteria. When we increased the amount of selenium, the size of the selenium didn't change, just the number. So we had more and more. And we got a patent for this technology in the States. And after that we started to develop different products with this new type of selenium. One product is on the right side, it is for feed and food additive, and on the left side for scientific purpose, just to produce pure nanoparticles. Actually, it was not really a nanotechnology because the size of these particles are 200 nanometers. And officially, the nanotechnology is below 100 nanometers. But the main questions were, what is the toxicity and bioavailability of these new products? So we wrote several articles about the toxicity, and we proved that it is not toxic at all. Anyway, we were not able to poisoning the test animals, and even it was an antidote of many other poisons, like polyaromatic hydrocarbons and even mycotoxins. If we add mycotoxin to the hen, together with the selenium, it had no negative effect at all. And we had many other test animals, like Paramecia. Paramecia, you know, in the one-cell organism, and when we gave the non-selenium, you know, the red one to them, they immediately run there and they started to eat it. They really liked it. And after that they collected inside in the cell. You see that yellow spots are the selenium. It is under the electron microscope. And even this type of selenium has a special effect on the plants as well. It is a tobacco plant, and we published something about the saving or extending the life of the plant. You know, there are two plants on that picture has the same age, but on the right side, which has the red media, it's much younger, looks much younger and much stronger, so it is a senescence effect. It means it keeps the plants alive longer, and even extremely high selenium concentration was measured in the plants without any toxic symptoms. Yeah, it is the article about that. And we tested this with animals as well, so when we gave it to animals, its bioavailability was much better than the organic selenium, and it was not toxic at all, because on the market we have the organic selenium and already we have this one. We wanted to understand the mechanism of these things what we found, because it's very surprising if something is not toxic at all and the bioavailability is good. Because if the bioavailability is good, it's easier to overdose, we consider. But in this case, we found a special mechanism and we explained that, what's happening and what is the reason. I don't go into details, but if someone is interested, I can explain this. And even we have a mechanism for availability. It's really true, and many of other research groups can follow the same things. In Europe, for the food, we can use health claims. Health claim, it means we can write it to the food. It is good for the health. Actually, in case of selenium, we have 6 health claims, what we can apply. Generally, we use these 3 on the packaging, because selenium protects cells from damage. It is antioxidant, and it is good for the thyroid hormones anyway. If somebody feel tired all the time and sleep too much, in that case probably has thyroid problems, and in that case the selenium supplementation is really good for her or for him. We have products already, a lot on the market with this product. We are selling this. The bottom, it is a thyroid complex, its name, like this. And even we put it to food as well. It is a very popular special dietary product in Hungary, and even it was advertised in the television as well. So that was the first story. We can earn some money from this. We're still studying this. We still have some interesting questions, but I'd like to talk about another story, which is not so old. It is only 2 years old. 2 years ago, I started and I had to set a lab experiment for students about the nanotechnology. We can produce and we can demonstrate the production of silver, gold, and many other nanoparticles and even selenium. And I realized some of the nanoparticles are very interesting. And in that time, we were dealing with the production of a special food product. It is called black apple and black banana. It is already on the market, we just started to produce it. It is a special technology which is suitable to make the banana black. It has a very good taste anyway. And we collected the black foods as well. And this nanotechnology meet our previous black food projects. You know the carbon has plenty of different interesting compounds. Not only the coal, what we can find in the mine, but almost all of the different form of the coal or the graphite or the carbon get Nobel Prize lately. You know the fullerene, not for the diamond, but the carbon nanotubes are very popular in that time, the production, and graphite and graphene oxide, so a lot interesting form, and lately they started to produce so-called carbon nanodots. And the carbon nanodots production connected somehow the food. I realized some people try to make carbon nanodots from food, just simply just with a heat treatment. And I realized we are dealing with this. We are dealing with these black foods, and it is black because of the Maillard reaction, and probably the Maillard reaction ends with carbon nanoparticles. So we tried this, we mixed sugar with amino acids, and we started to heat it up, and the color changed to brown, and we realized it is really seems nanoparticles, and finally we discovered that it is not exactly carbon nanodots, we can produce it. It is a new nanoparticle. We give a name for this carbon seed nanodots because it looks inside, it has a seed, a core, which is probably carbon, and around these we could put plenty of different materials like amino acids, even EDTA, or bioactive compounds like curcumin, or different spices or something. If you put this liquid, what we produced in the previous experiment, to paper, and you use a UV lamp, in that case you can see at the border, you have a very nice and strong fluorescence. Because the carbon seed nanodots strongly fluorescent, I will show it to you in this water. Before my lecture, I put some to this water. I drank some from this, so it is edible anyway. And if we check the different food, what was totally not known before, some of them are really full of this new type of material, this carbon seed nanodots. It is the coffee, you know, the coke, and even activated carbon, and we can identify it easily with the UV laser. You can see the fluorescent effect. We measured the size of this particle by a transmission electron microscope, and we can check the fluorescence of this. The size of the particles is about 3 and 5 nanometers, so it's really small. And we were thinking a lot how we can separate it, how we can purify it, and how we can measure it. It is an example for the purification. You see the glassware has really nice color there, and we measured the concentration. We made a standard for this new material, carbon seed nanodots, and we compared the amount of different food in there. So the coffee contained the most from this, and it is 3,000 milligrams per kilogram. And actually it's a lot. And we didn't know about it at all. All foods full of this type of material. Yes. In this direction. And even if your wife make a chicken at home for dinner, try to use a UV lamp like this. And if you highlight it, it will have a nice blue fluorescence, like this drink. I put some carbon nanodots or carbon seed nanodots to this drink. Anyway, it's edible, it's drinkable, so it seems very, very interesting material. After 1 year thinking, we found the best method for the separation and the measurement. And we found that HPLC you know, the high-performance liquid chromatography connected with the size exclusion chromatography, which separate the particles according to the size, and equipped with the fluorescent detector. It could be, and it is a good way for measuring these carboxyl nanodots from the food and from everything. And we have experiments with plants and soil as well. They are very exciting and promising. And anyway, if you see these chromatograms, the first two peak, definitely carboxyl nanodots, because we know the size of this. Its molecular weight is, the first is 700 or 800,000 Dalton, and the second one is 350, and the last one is only 600 or 700. So the 700 is a molecule, or molecule-like. We don't know exactly what is that. It has a chance it could be really carbon, probably fullerene, but I'm not really sure of this. But the first two definitely this type of new material. And this size of particle is much bigger than a molecule, much bigger than a protein or something. It is real nanoparticle. We published it in many journals, and we have that plan to produce it in a high scale. We can produce it in larger amount. So if anybody would like to take part in research with us on this new material, if we publish together and if we work together, we can send free for you. If somebody would like to make a product, of course, it will have a price once, but not in this moment. So thank you for your attention. If you have any question, I don't know is it legal or not, but you can.
Any questions?
Thank you so much for this presentation. Please. What? Please, I need to have maybe number 5 or 6 slides. Structure of you know about carbon tube. Yes, you. So please inform us about this formula. This carrier, this used as a carrier. This structure. This structure. Carbon nanotube. Yes, yes. You can use as a carrier for plateaus.
Yeah, it's okay. Officially, you cannot use it in food. Because, you know the problem, if you produce something new, in that case, that is called in Europe novel food and novel compounds. But the biggest advantage of our product in this case, we don't produce it separately and add it, but we realize it is normally in the food. So we can find such a normal food which rich in that. So we will start, actually my PhD student started to eat a food which rich in this material, and she took the blood samples, and we will make a bigger clinical trial on this, because it's legal to do this, because it's a natural material, and the legislation would be much easier if you can prove that already it is around us, we eat it, or we ate it in the last some thousand years. So in that case, it's much easier.
Without any side effect?
Of course, we will make experiments and we will make animal experiment with the purified samples. We will make human study and clinical trials with that food, which is knownly rich in this material. And lately, we started to measure these nanoparticles in our body. We measured it from mice already, so we gave this to the mice, and it was detectable by this method in the mice, the different part. So we're just producing curcumin nanoparticles with the same method, and we wonder it goes to the brain or not to the mice, because it's really challenging.
Another way, we can use the biosilica extracted from diatomies.
Yes, yes, I know this.
Yes, yes. Thank you.
Thank you.